Pure culture technique of microorganisms. Division of a single bacterial cell results in the formation of a pure colony. What is culture? The microorganisms that are cultivated is known as culture. We have two different forms of culture, that is mixed culture and pure culture. In mixed culture, we can see more than one microorganism. In pure culture, it contains a single species of microorganism. How do you assess the purity of culture? Several isolated colonies of a pure culture must show the same cultural characteristics on a particular media. When they are stained and observed under the microscope, they look alike, that is, same stain and morphology must be seen. Several isolated colonies perform identically, as in the case of similar biochemical results. Cultural and morphological characteristics of mixed and pure culture. You can see the differences in the image. Mixed culture colonies on a petri plate and pure culture colony on a petri plate, and the lower one is the microscopic field of the organism. These are the pictures showing some biochemical tests results. Mixed culture showing different results and pure culture showing similar results. When we conduct a series of biochemical tests for your pure culture, it must show the similar results. What is the importance of pure culture? Pure culture can be grown, identified, characterized and tested. The physiology and the clinical aspects can be studied. For a particular culture, same results are obtained irrespective of the number of times the test has been done. Every time we should get the similar or same result for pure culture. The rate of spontaneous mutation is slow and the clone of a pure colony is identical in all aspects. Pure culture technique was first adopted by Robert Koch. He isolated Bacillus anthracis for the first time. Bacillus anthracis is the causative agent of anthrax. What are the techniques involved in pure culture? Sterilization of media and glasswares. Sterilization is attaining zero microbes in the glasswares and media, dispersing the individual cells across the medium, thinning the samples many times before inoculating the fresh media. Sterilization techniques, we have moist heat sterilization, dry heat sterilization, filtration, flame sterilization, biosafety cabinets, that is laminar airflow. These are the different sterilization techniques involved in pure culture technique processes. In the picture you can see autoclave, that is used for sterilizing your glasswares, media and other apparatus used for the inoculation techniques. Hot air oven, that is dry heat method. Moist heat method. Sitz filter for filtration purpose. Flame for incineration of your loops, needles, etc. and to conduct your inoculation techniques under aseptic condition. Laminar airflow, this is the laminar airflow cabinet which gives 99.9% germs free area for us to conduct all microbial techniques. Isolation of pure culture. For isolation of pure culture, we have different methods such as streak plate method, pour plate method, serial dilution technique, spread plate method, and enrichment method. Streak plate technique. All these isolation techniques have been already taught for you all in your practical classes. Now we'll just see the overview of all these techniques. Streak plate technique. How to do it? We take a sterile Petri plate containing sterile medium, allow it to solidify, then from pure culture samples we'll take a loop full of sample in a sterile inoculation loop and streak it on the surface of the solidified agar on your Petri plate in zigzag manner or parallel, radiant, etc. That is about streak plate technique. These are the various streaking methods. Then we have pour plate. For pour plate method, we have to add small amount of inoculate, that is organism culture, into a Petri plate, sterile Petri plate, and then we can pour the medium, mix it and allow it to solidify, then incubate it to see the colonies. Or else you can pour the medium first into a Petri plate, allow it to solidify, then you pour your sample, that is culture, mix it and incubate it to see the bacterial colonies. Serial dilution technique. For serial dilution, we take series of test tubes, and to the first tube, in all the tubes we'll take 9 ml of distilled water or saline. To the first tube, we'll consider it as 10 raised to -1. We'll add 1 ml of starter culture, 1 ml of sample containing organism of interest. We'll mix it and again we'll take 1 ml from that particular tube to the adjacent tube, that is 10 raised to -2 tube. Same thing we'll do till 10 raised to -10 tube to get the dilution, serial dilution of the particular culture or organism. After conducting serial dilution, we'll take each tube as a sample and conduct plating method, any of your inoculation plating methods such as streak, pour, spread plate, etc. Using that serial diluted samples as the inoculates. After incubation, you can see the colonies where 10 raised to -1 tube shows more colonies and compared to 10 raised to -10 tube. Colonies will become lesser with it when dilution increases. More the dilution, lesser the colonies. Spread plate method. We'll take the sample, 0.1 ml of sample is added to a solidified agar medium, nutritional medium. Then with the help of a spreader, we have to spread the inoculum, then incubate it and observe the colonies. Everything must be done under aseptic condition. Enrichment method. Basal medium is incorporated with blood, antibiotics, growth supplement to enhance the growth of the desired culture. Once the pure culture is isolated, maintenance and preservation is necessary for the further study of pure culture and the application of the culture.